and she received bachelor's degrees in both chemistry and mathematics. And then she made the wise choice of choosing chemistry and as the subject and UCI as the location of her graduate studies. Given her impressive credentials and a positive impression that she made when I first met her after arrival in fall 2015, I was very happy that Vicki asked me to sponsor and suggest a project for her application for the National Science Foundation's Graduate Research Fellowship Program. Her application was successful, and the fellowship supported her contributions to a fruitful collaboration between my research group, the Mobley group, and the Tombola group. It also enabled her to do a 3-month trip in Stockholm, Sweden, where she had the opportunity to engage in cutting-edge structural biology research. So Vicki did a lot of work at UCI in the last 5 years, spanning a broad range of topics. The general theme of developing and applying techniques for using computational modeling to model the dynamics of biological molecules and use the output of the modeling in conjunction with statistical mechanics theory to elucidate pharmacological principles. So I think now I'll turn it over to David Mobley, Vicki's co-mentor, and he can comment on his experience working with her.
Yeah, thanks. Thanks, Doug. So, I think it's been great. It's been great working with Vicki over the last few years. She's extremely capable and competent at everything she does. Really, really excellent, excellent work. And it's interesting. She came with quite the diverse research background, including in organic synthesis and doing computational chemistry internship at Merck before coming here. And so I think that's meant that she's really been able to tackle a set of extremely diverse projects, which is great. And in addition to these diverse research projects she's done here, she's also done a lot of other interesting things on the side in terms of, including quite a lot on scientific communication and outreach, getting involved with tech transfer. I would say probably more than an average graduate student in terms of lots of other interesting outside activities. And so I think that's led partly into her becoming interested in pursuing a career in the patent area. But she's really been a delight to work with. And I'm looking forward to hearing from her again today and staying in touch throughout her career, which I'm sure will be excellent. Just a couple of quick logistical details before we get going. I was asked to remind you of that there's a preference that audience member mics muted and video be turned off during the formal talk. Hold your questions until after the public talk is concluded. Then there'll be a question and answer time after that. If you want to ask a question, you can type into the chat saying that you have a question, and then you'll be called on at the right time. The moderator will unmute your mic at that point so you can ask a question. And then afterwards, the committee and Vicki will leave for a bit to a different Zoom meeting to discuss with her, and we'll leave this one open. And then we'll she'll return to this room at the end. So if you want to stay in this room for a bit at the end, you'll be able to congratulate her afterwards and interact with her more at that point. So I think that's it. So Vicki, you should be able to get going.
Okay, wonderful. Let me share my screen. Okay, does everybody see my title slide? Okay, great. So thank you all for being here today, and I'm really excited to share with you my research in the area of computer-aided drug design. Okay, so we're going to start with a drug that we're probably all familiar with, which is aspirin. Aspirin is a small molecule drug, as are most drugs on the pharmaceutical market. And these small molecule drugs often work by tinkering with some protein in our body. Proteins are these tiny molecular machines that keep our body running at the most basic level. Our body has thousands and thousands of proteins, but they don't all work as expected, which can lead to various afflictions such as pain and cancer. So with the general idea that we can tinker with a protein to treat disease, some questions arise. Specifically, how do we know what protein we need to tinker with? And then how do we design a molecule to tinker with that protein? And even if we design the perfect molecule, how do we know that it will even be able to reach its protein target? Now, these are complex questions, and drug discovery in general is a complex process, taking on average 10 years and over $2 billion to develop a new medicine. But we can use computer simulations to facilitate that process. So in simulations, we can build models of proteins and potential drug compounds to really look at the specific interactions between them. And this level of insight is difficult to obtain in molecular from experiments. So how do we build these models? Well, we represent atoms as spheres. So in this water example, the sphere for oxygen will be bigger and heavier than the spheres for hydrogen. And we represent the bonds as springs. So from physics, we know that the energy of a spring is represented by this harmonic potential. And there are two constant parameters. One, K, that tells us how stiff the spring is, and one, R naught, that tells us the resting length of the spring. But we don't just have bonds in a molecule. We can also have angles, dihedral angles, improper dihedrals, and different types of non-bonded interactions. And just like we had a formula for bonds, so we also have formulas for the different other interactions. But you don't have to pay too close attention to these formulas here, because what I want to emphasize is that like we had the constant parameters for bonds, we also have the constant parameters for these other terms as well. And the set of equations, as well as the different parameters, so different constants for the different types of bonds, this makes up what we call the force field. And I'll be referring to this term throughout my talk. So now that we have a way to build the model, how do we actually look at the motions and interactions that we're interested in? We just use Newton's second law. So I showed you that we have a way to express the energy of a system. Then we can use that to get the forces by taking the negative gradient. And then we can apply that into the second law, F equals ma, to get the updated positions and velocities over time. So this is a process known as molecular dynamics. So we can use this to predict the motions of these individual atoms to analyze their interactions. An example of this comes out of D.E. Shaw research, where they're looking at a potential drug molecule to bind to this human protein. And this protein is actually what lets SARS-CoV-2 into our cells. So this is a potential treatment to address COVID-19. So with this background, I'm going to transition into what I've been focusing on over the last 5 years. So these are a number of different works that I've done throughout my grad school career. And just in case you couldn't read all that, I also have a pictorial representation where the top row represents primarily first or second author contributions. But in the interest of time, I'll just be talking about the first 3 projects. So first, we'd like to talk about binding free energies. So you remember in the beginning, I talked about what protein do we want to tinker with. Well, there's been a lot of experimental studies suggesting that H3.1 would be a good potential therapeutic target. So H3.1 is a voltage-gated proton channel. It's a protein that lives in our cell membranes, and it's responsible for protons to go out of the cell. This process contributes to the production of reactive oxygen species for our immune system, and also contributes to the bioluminescence of dinoflagellates and also the maturation of sperm cells. The reason we're interested in this for a therapeutic reason is that it's been known to be associated with the metastasis of breast cancer, as well as neurodegeneration and ischemic stroke. So now we want to think about how we can tinker with this protein. And the Tombola lab here at UCI has done experimental work on this group of molecules, guanidine derivatives, that have been known to inhibit H3.1 to varying degrees. So my focus will be on compound number 1, whose name is 2-guanidino-benzimidazole. I call that 2GBI. And we're not just looking at 2GBI itself, but we're looking at two different structures of 2GBI, two tautomers, where a tautomer is basically having the same chemical formula, but it differs in the placement of a hydrogen atom. So we see that the shaded blue regions represent more positively charged regions that depend on that hydrogen atom. And different tautomers might be able to have different interactions with a protein. So my goal here is to evaluate the binding of 2GBI to H3.1, and we're also looking at H3.1 mutants. So in order to evaluate the interactions between the ligand and the protein, we need to have a starting model. And a lot of models often start from experimental data, like crystal structures. There is no crystal structure of human H3.1, so the Tobias lab previously developed a homology model using a mouse chimeric construct. So using this model and the ligands, I want to explore various questions. Specifically, what interactions does 2GBI form with H3.1, and will that differ by tautomer? And then secondly, can we compute the binding free energies and compare them to experiment that will validate this model for future studies as well? Okay, so the first thing we need to do is see potential placements of the ligand inside the protein. And we do this using a process known as molecular docking. So docking gives us a various number of configurations of the ligand and the protein. And we look at those one by one and evaluate them with respect to experimental data, looking for residues that are known to play a role in binding the ligand. So with that and refinement in MD simulations, we have a pose that is shown on the right-hand side where we identify a few key residues that were known to be involved in binding from experiments, but also we identified a few more that might be important too. So then after this, now we want to turn our focus to computing the relative binding free energies. So this is a thermodynamic cycle based on the principle that free energy is a state function. In other words, it doesn't matter the path you get to A or B, it just matters what is the energy difference between the two. So here I'm showing the wild type protein with the specific residue in question in purple, and then I'm showing the mutated protein with another set of atoms in green. So experimentally, what's measured is delta G1 and delta G2, but this process is hard to simulate in our MD simulations. So we use a technique and actually calculate delta G3 and delta G4. And we use an alchemical approach. So what that means is that we're actually having both sets of atoms, the initial protein residue and the mutated protein residue, inside the system at the same time. But we can't simulate them directly because oftentimes the relevant states that one will sample is not the same as the relevant states that the other will sample, and so that will lead to unconverged results. So the way we address that is that we introduce these alchemical intermediate states, where we take state A and we turn off its interactions slowly over time, and then we turn on the interactions of state B slowly over time, so that we have overlapping phase states along the way. So even though we have these intermediate windows, there's a number of things that can contribute to effective or non-effective simulations. So specifically, the focus here is on flexible charged side chains, specifically arginine or lysine. And so what we observe in simulations is that these residues are able to flip outside the protein, so it's actually in the membrane region, whereas it's invisible and not likely to be in real life. So the way we address that is apply distance restraints to that charged residue to the surrounding glutamate residues. So ordinarily, this residue would be interacting with these acidic residues, but is not able to when we turn off the interactions. Secondly, we might see differences in hydration patterns. So as I mentioned earlier, free energy is a state function. So when we take the energy of A to B, it should be the same as the energy from B to A. And so what I looked at was a different mutation where I'm mutating arginine to serine. And when I mutate A to B, which I'm not showing, and then back to A, I saw that I don't actually get A. I get a different state, which is A prime. So that's what's being represented in the blue spheres, which are water molecules. We see that the water network around the mutating side chain is not the same as what we started, and that actually leads to differences in the AB to BA energy. So we see that in cases like these, we don't have very conclusive results. So with all that being said, these are our computed relative binding free energies. So I'm showing here 6 protein mutations that are colored in the different colors. And I'm also showing both tautomers, the GBI1 in circles and GBI2 in squares. And all this represents over 8 microseconds of simulation time, which is a considerable amount. So what we see is that generally this, the solid line that's sort of diagonal represents the perfect agreement with experiment. So we see that the circles are further away from the solid line than the squares are. So this suggests that GBI2 might be the more prevalent species in binding HV1. But overall, we see qualitatively pretty good agreement with experimental data, which really supports our model of HV1. So to conclude this section of the talk, we saw that with refined docking, we can generate reasonable configurations with consideration to experiment for TGBI bound to HV1. We see that conversions may be affected by charge flexible side chains or hydration patterns, where we were able to address the side chain issues using distance restraints. And then finally, we see decently good qualitative agreement with experimental results, and this supports our open state model of HV1. So now I want to move on to looking at membrane permeabilities. So in the beginning of the talk, one of the questions I raised was, how do we know that the drug that we design will even be able to reach its protein target? And so this is where membrane permeability comes in. So even if we have the perfect drug, the drug will have to pass through at least one membrane to reach the protein target. So poor membrane permeability often means poor efficacy in vivo. So our cells are surrounded by a lipid bilayer that is selectively permeable. Some things will be likely to permeate, whereas others won't. So the things that will be likely to permeate are small hydrophobic compounds, whereas polar compounds will sometimes permeate with a preference for smaller, uncharged things. And then finally, these ions that have a concentrated charge will be not as likely to permeate passively. So here I'm introducing a new molecule. We saw in the last section 2-GBI, but here now I'm introducing 5-chloro-2-guanidino-benzimidazole, which I call chloro-GBI. So this is different than 2-GBI by the placement of a chlorine atom, which was a hydrogen atom in 2-GBI. And what's really interesting about these is that 2-guanidino-benzimidazole is known to block the HV1 channel from the intracellular compartment. When it's in the extracellular compartment, no block is seen. But 5-chloro-2-guanidino-benzimidazole is able to inhibit HV1 when placed in the extracellular compartment. And they are believed to have similar mechanisms of block. So the hypothesis here is that chloro-GBI permeates through the membrane and then accesses HV1 from the intracellular side. So this is the hypothesis that we want to test. So we do that in simulations using what is known as the inhomogeneous solubility diffusion model. This gives us an expression for the permeability coefficient PM. This is an experimentally accessible value. This depends on 2 contributions, the diffusion coefficient, which is the rate of mixing, and the potential of mean force. And we see that there is an exponential dependence on the potential of mean force. So you can think of this as the free energy surface projected onto a transition coordinate. So in my case, as I'll describe, the transition coordinate is one-dimensional. So you can think of this as a one-dimensional free energy curve. So as I mentioned, we're looking at 2-GBI, and here we're exploring the 2 tautomers as well as chloro-GBI. And this is the setup of my membrane. So I plotted it sideways so you can really see the different contributions of different parts. So we have the POPC lipid molecules that comprise the membrane, and you can see that there's different parts to the lipid molecule, and the different parts are shown in the different colors in this plot. So we see water here, and then we can see that the different contributions lead to a very heterogeneous surface, so that there's going to be a lot of complexity as the solute permeates. And then we define our one-dimensional coordinate as just the distance from the center of the membrane, as shown on the bottom. And the individual rectangles, the blue and red rectangles, represent how we stratify our simulations in order to facilitate efficient sampling. And these are what I call windows. Okay, and we do our simulations using a technique called adaptive biasing force. So a common challenge in molecular simulations is that we will typically just prefer to sample the low energy configurations such as here. And it's actually pretty difficult to surpass these high free energy barriers to actually get a sense of the whole space. So what is done in adaptive biasing force is that we apply a biasing force that is updated over time, and that translates to a potential that exactly cancels out the barriers that are seen by the solute. So that leads to a flat free energy surface, which provides an ideal sampling so that the solute can sample the whole space. So another simulation detail that I need to explain is that we approach the potential of mean force bidirectionally. So the reason we do this is that it can be hard for the polar groups of the solute to really sample the interior of the membrane. And so what I'm showing in the left side is that a solute might be associated with the lower leaflet at one time point, and then it flips over to the upper leaflet. And it will stay there for some time before it flips over again to the lower leaflet. So this is almost 300 nanoseconds of simulation, which is pretty considerable. So in order to facilitate efficient sampling, we actually compute the potential of mean force from the upper leaflet, which is shown in green here. So we take those windows, and then we also do the same thing for the lower leaflet, and then we can just combine them because configuration integrals are additive, so we can just make use of that statistical mechanical principle. Okay, so with all of that, the potentials of mean force are shown here. And so here, GBI1 is shown in green and GBI2 is shown in purple. And what is sort of most noteworthy right off the bat is that we see chloro-GBI, which is shown in blue, and it has the lowest potential of mean force of the three. So this suggests that from a thermodynamic standpoint, GBIC is the most favorable to enter. And we also compute the partitioning free energies as from integrating the Boltzmann-weighted PMFs. And we see that the partitioning free energy for GBIC is lowest in energy, so it will be most likely to enter from a thermodynamic standpoint. So in order to explore more of what happens during these simulations, we took a closer look at the membrane. So here on the left side, I'm showing what does the membrane look like when the solute is in bulk water. And right below that, I'm also plotting the electrostatic potential plots. So here you can think of that as sort of a free energy surface, but for charge. So we see that when the solute is in bulk water, there's not really any effect on the membrane. It's pretty flat and normal. But when the solute is in the interior of the membrane, basically the interior of the membrane is hydrophobic, and there are some hydrophilic parts of the solute. So what actually happens is that it's affiliating with different parts of the lipids, and it also coordinates with some water molecules that leads to considerable distortion of the bilayer. So we see that most clearly in the electrostatic potential plots, as in the lower right-hand corner. So this is shown for GBI2, but we also notice this effect for GBI1 and GBIC. So then we turn our focus to what is actually happening with the solute as it permeates the membrane. So the way we do this is by assigning a vector from the carbon atom shown on the bottom to the nitrogen atom shown up top here. And we define the cosine similarity score with the z-axis, where the z-axis is normal to the membrane. So a value of +1 means what I call pointing up, and a value of -1 is what I call pointing down. So we do this and we compute this for all the windows that I mentioned that we have. And so here what we see is that we're first going to take a look at the purple windows, where the solute is in bulk water as shown on the left here. So here it's an isotropic medium, meaning that the environment is the same on all sides, so it doesn't prefer one orientation or another. And then if we skip to the green here, we see that there's sort of peaks on both ends of +1 and -1. So basically what happens here is that we see a lot of NH groups on the tail of the solute. So it's actually interacting with the head groups of the lipids, while the benzene group is in the water. And then at some point around here, it flips around, and we see this strong preference for +1 where it's pointing upwards. And this makes a lot of sense because we have a very hydrophobic interior, and we also have benzene, which is a more hydrophobic moiety of TGBI or chloro-GBI. And so it's wanting to basically like dissolves like, we're having similar parts in similar parts of the membrane. But something that's interesting to us here is that the effect for chloro-GBI in the lower right-hand corner is most extreme compared to both GBI2 and GBI1. And so to explore that a little further, we zoom in and take a look at those interactions in our MD simulations. And what we see here is that, like we predicted, the NH groups of 2GBI are able to interact with the negatively charged phosphates groups of the lipids. But what we have in addition is that this chlorine atom can be solvated by the hydrocarbon tails. And a chlorine atom is much bigger than a hydrogen atom, and so it'll also have a lot more dispersion interactions. So we believe that the addition of these dispersion interactions contribute to the strong orientational preference of chloro-GBI, which also may affect why it is more likely to permeate than 2GBI. So to conclude this part of the talk, we see that chloro-GBI has a lower energetic barrier to membrane permeation, and which is consistent with the experimental observations of chloro-GBI being able to block HV1 from the extracellular compartment. Secondly, we see that all permeation events lead to a lot of distortion in the membrane bilayer. We also saw that solutes have varying degree of orientational preference based on their location as well as based on the chemical composition. And then finally, we think that these dispersion interactions are what contributes to the stability, relative stability of chloro-GBI in the head groups compared to the various tautomers of 2GBI. Okay, so with that, I'm going to move on to the third part, which is on force field evaluation. Okay, so in the beginning, you heard me talk about small molecules tinkering with proteins to treat disease. So if we have some small molecule in a protein binding site, we need to in order to make sure we have accurate assessments of the system, we might need to consider a few things. So specifically, what geometries can that molecule adopt? And are these geometries physical and are reliable? And then secondly, what are the energy differences between them? So, things in nature will often prefer lower energy states, so we might be able to predict which one is a more stable binding compound compared to the other. So this is where I'm evaluating force fields, which are those, once again, the parameter sets and the functional forms that are used to model our system. So we're looking at several different force fields. So let's say we have some reference structure where the reference comes from quantum mechanical data. We're looking at the Amber family of force fields, GAF and GAF2, the Merck molecular force fields, 94 and 94S, and also two force fields, Parsley and Smirnov 99 Frost, which come from the Open Force Field Initiative. And Smirnov 99 Frost is actually a descendant from Parsley, so we would also hope to see that there's improvements in Parsley compared to Smirnov. So as a high-level view of this workflow, I extracted the molecule set comprising over 26,000 molecules from QC archive, and I grouped the conformers based on, so what is shown here, I grouped the conformers based on connectivity. So everything with the same connectivity is considered a part of the same molecule, where we have different conformers of a molecule. And then we have this reference QM data, which I'm showing in blue, and we perform, using that starting structure, we perform energy minimizations with the different force fields that are shown in these three colors, the teal, purple, and yellow. Well, we're doing that for all 6 force fields. And then finally, we compare the force field data against the QM reference, looking at those geometries and energies. So first, we're going to focus on their energies. We compute the change in conformer energy using this formula here. So essentially what we do is we assign a reference conformer for every molecule, where the reference conformer comes from the lowest QM structure. And then for the rest of the conformers, we take its change in energy relative to that. So we'll have DE1 and DE2, etc., for all the conformers. And then we just take the force field result and we subtract the QM result. So ideally, if we have great agreement with QM reference data, we'll have a lot of zeros, meaning that the change in conformer energy for force field matches the change in conformer energy for QM. So these results are plotted in the histogram on the right-hand side, and we see that, yes, we do have a lot of values around the 0 bin. Generally, a lot of the force fields look pretty similar in their relative energies, relative conformer energies, but the 3 force fields that tend to do a little bit better than the rest are GAF, GAF2, and parsley, which overlaps with GAF. And we also see a slight asymmetry in this plot in terms of the reproducing QM conformer energies. So given the fact that the same starting structure in QM can minimize to different final structures from the force field, we also want to consider matching structures before we evaluate the energy. So what we do is match all the structures within 1 angstrom RMSD of each other, and then compute their change in relative energies. So the mean sign deviation formula is similar to the DDE formula I showed on the last slide, but now we're just considering matched conformers. So these results are shown in these violin plots. And so what we see here is that the white dots are the medians. And once again, it's a little bit on the negative side, so reflecting that same asymmetry that we saw earlier. But generally, we see that the distributions, once again, are pretty similar for all the force fields. But just to take a closer look at any differences, we zoom in to these violin plots and then overlay them together, which is shown here. And so what we see is that if you look carefully on the inset, we see that the purple, blue, and orange tend to be wider, meaning that they have the deviation, a lower deviation, meaning that they match QM better. So they tend to reproduce these energies like we saw in the histograms. So now we're going to turn focus over to geometries. Okay, so we evaluate their geometries using 2 different metrics, RMSD and TFD. So RMSD is root mean square deviation, and it's more commonly used when we're comparing different structures. But RMSD is size dependent, meaning that we might have larger values of RMSD just because we have larger molecules that we're comparing. So we don't want to just identify larger molecules, we want to identify molecules that are different. So that's why we introduced TFD, which is torsion fingerprint deviation. So the TFD was designed to be more size independent by taking into account the Gaussian weighted torsion differences. So using both of these metrics, we histogram them. And so the values of RMSD are lower bound from 0, meaning 0, so a number closer to 0, meaning that the geometries are in very good agreement, and it's not very limited. It's unbound in terms of its molecule size. And TFD is normalized from a scale of 0, where we have good agreement, to 1, which is less good agreement. So these results are histogrammed here, where we also apply a kernel density smoothing function. So we see that in general, the purple and brown tails are a little bit bigger than the other force fields in both the RMSD and the TFD metrics. But we see that parsley is an improvement from Smirnoff 99 Frost, which has a bigger tail and it's more skewed over to the right. But if we look at the bottom plot of TFD, where now we control for the molecular size, we see that MMFF 94 and 94S force fields tend to do a bit better in reproducing the QM geometries relative to the other force fields. So we can use this geometric measure to identify parameters that might be improved upon for future force field development. So what we do is define this overrepresentation ratio, where we look at all the molecules that are found in the tails of the distributions just above some cutoff, and then we look at all the parameters that are in that molecule. So in this case, the bond parameter B83 might be repeated a number of times, but we'll just count that as 1 time. So we look at how many times we'll see that parameter in just the tails, and then finally also in the whole set. And then we compute the ratio of the tail to the whole set. So a higher number means we're more likely to find it in the tails compared to just the rest of the distribution. So we're wanting to focus on high overrepresentation ratio parameters. So this is plotted in something like this, where I'm showing the bonds and angles and van der Waals interactions. And the purple dotted line shows a fraction value of 1. So you can see that there's a number of parameters of all of these that have numbers significantly higher than 1. So these might be a point of focus when we're working on improving the future force fields. So now moving on from geometries, now I want to look at the interplay between the energies and the geometries. So I'm plotting that as a scatter plot shown here. And so we've already seen the 1D versions of this data, but now we're just looking at the interplay. And so the coloring here represents density. So a region of blue is high density. So we're actually plotting over 20,000 points on this. So we're trying to get a sense of how compact these points are. And red regions are areas of lower density. So once again, with a perfect match between the force field and QM, we would have a high density of points around the origin, 0, 0. And what we're looking at just for parsley here is that there's not a direct relationship between the geometries and the energies in terms of relevance to, in terms of reference to QM. We also, instead of looking at just parsley, we look at all the different force fields here. And so you can see that just from a bird's eye view, they all look pretty similar, but Smirnoff 99 frost is more spread out in distribution than the rest. And this is consistent with what we're seeing in the histograms where they were also more spread out. And it's a little bit hard to see the trends on a linear scale, so we also plot the same thing on log scale. So here we can see differences a little bit more clearly, where if we compare GAF2 on the top right to GAF on the left, we see that the high density regions becomes a little bit more symmetric with the newer version of GAF. And the same thing is true for MMF 94 and 94S, where we have this blue region becoming a little bit more symmetric about 0, 0. And then with parsley and Smirnoff 99 frost, we see we do have dense regions around 0, 0, but Smirnoff 99 frost is much more spread out, as we're showing in the lower right corner. So in addition to looking at these, we want to also identify molecules that might be useful for improving future force fields. And so that's what I'm showing in this plot here. So here I've identified 3 moieties that might be areas of improvement, specifically the NN single bond compounds, which tend to be pretty spread out, as in the teal here. We also have the azetidine compounds, which have this 4-member ring with a nitrogen, which accounts for a lot of the high deviation in geometries with the high TFD scores. And then finally, we also have this octahydrotetracene structure, where this scaffold is primarily responsible for a lot of these energy differences relative to QM. So with that, I'd like to conclude this part of the talk. So here, we saw that most force fields are comparable in energy, but we have slightly higher agreement with QM with GAF, GAF2, and parsley. We see that parsley improves from Smirnoff 99 frost in reproducing the QM geometries. We see that there are distinctions in the high density areas between different force fields. And then finally, we identify specific moieties that might be areas of improvement to develop future force fields. And the code for this setup, analysis, and everything is available on GitHub. And so if you're interested in force field evaluation, feel free to check that out and let me know what you think. Okay, so this concludes the research I'm presenting today. I would like to thank my PhD committee, David Mobley, Doug Tobias, and Rachel Martin. David and Doug were my co-advisors over the last 5 years, and I've really learned a lot working with them, and it's really been a privilege. I'm thankful to Alfredo and Francesco. Alfredo has been this endless source of wisdom in terms of setting up and analyzing molecular simulations, and just gives a really good feedback overall, and has made a lot of my writing and work generally better. Francesco's lab did a lot of the experiments that I refer to in this presentation, and he's just been a very valuable source of insight for understanding the biophysics of this system. There's also a lot of individuals that contributed to various projects that I am grateful for. And I'm also appreciating funding from primarily the JRFP and some from the NIH, and also with the GROW Fellowship and funding from the Swedish Research Council to spend a summer in the Lindahl lab in Sweden. I thank the Tobias lab, especially Eric Wong, Andrew Geragatellis, and Michael von Darmros. So Andrew and Eric helped me get up and running on these molecular simulations, and Michael had a lot of useful insights regarding the permeability work. And also shout out to Ellie for being the funniest member of the Tobias lab. I'm super thankful for the Mobley lab, affectionately known as the Mobsters. I've gotten close, and special thanks to these individuals who I've gotten close to over the years or were integral in some of my projects. It's just, I've really enjoyed the company of the entire Mobley group over the years. I'm had the opportunity to do a lot of different things while at UCI, which really contributed to a valuable graduate research experience. So I appreciate Fong Long and Bree McWhorter for a lot of the science communication stuff and just personal development during this graduate time, and also others in various communication or other capacities. I'm thankful to the Lowdown on Science. They're a radio show and podcast, so if you're interested in science and humor and go check it out. It's really relatable. So specifically, I'd like to thank Sandra and also some co-managing editors on that front. I'm thankful for the Irvine Graduate Student Ministries and especially the sisters there who have just been provided like essentially another family to me during my time in grad school. And then I also am thankful for Applied Innovation, where I had a technology transfer fellowship, and that really showed me to think about science in a different way. So not just the research, but how can this be protected in terms of intellectual property and marketing. Finally, I'd like to thank my family. So this picture here was taken almost 3 years ago today when my brother graduated from pharmacy school. And my little sister on the right side is in med school right now, so soon we'll be a family of very different doctors. And I'm very appreciative of my parents, Tran and Lee. They've just been a huge source of support and inspiration and love over the past, just throughout my whole life, and I'm really thankful for them. I'm really grateful to my boyfriend, Justin Smith. He's been by my side throughout most of my grad school time, and just been just a light in my life. His sense of humor, his calm demeanor, and his just love and support have really taken me a long way, and I'm very appreciative of his being there. And then finally, I would just like to say grad school can feel like a very crushing process at times, as this fellow probably relates. But I would say, I just thank God for all these relationships and the friendships that I've formed throughout the years, and really just the strength and the perseverance to get to where I am today. And thank you.
Thank you.
Thank you, Victoria. Nicely done. So we're going to open up the question portion. So if you have a question, you can message the chat, and then I'll go ahead and unmute your mic.
Well, people are thinking, I can start off with one. Was there anything you found particularly surprising in the permeability work that you were doing in terms of your findings for the different 2GBI variants that you're looking at?
In terms of the chemistry, not as much surprising, but I think what was more surprising was just the different methods that can lead to different results. So I also tried another technique, which was a variant of ABF, which was meta-EABF, and that just, the results were similar but different, and so it was just interesting to see like, oh, what is the range of errors that we can get? So how reproducible are these results, and sort of can we get an estimate from different techniques of the membrane permeabilities of these compounds? So to answer your question more succinctly, the surprising thing was the lack of reproducibility from different methods.
So I have a question about your small molecule interacting with the membrane. So I thought it was interesting that you got so much distortion of the membrane in the vicinity of the small molecule. Mm-hmm. How localized is that? And have you done any playing around with how big you can make it? So in other words, can you make a small molecule that introduces membrane distortions and interacts with signaling proteins that measure membrane curvature?
So we didn't explore how big the distortion was. What we did in that regard was to make the membrane big enough such that the edges wouldn't be distorted, so that when we're taking into account periodic boundary effects, so that we have a more uniform system. Um, we've past literature also shows that a small water molecule permeating will also cause some distortion of the membrane, but to a lesser degree than these compounds. So I imagine if, for example, if we had some of these, a steroidal compound with a polar group on it, that is bigger and also has that polar group. So I think that would cause even more distortion than our relatively small 2GBI. And yeah, I imagine that could affect some of the proteins that surround it in the membrane.
Cool.
I have a follow-up to that. So the three compounds that you considered for permeation were all positively charged.
Mm-hmm.
And I suppose that you've looked at the literature for various compounds permeating membranes, and is the extreme barrier that you found for the non-chlorinated compounds, is that unique to the molecules you considered, or is it more generally found in other charged compounds? And is there a difference between positive charged and negatively charged compounds?
Yeah, so I've seen some studies in the literature, I think by Tobias and coworkers, where they looked at specific ions. So they actually pull these small charged ions through the membrane. And in those cases, they saw considerable free energy barriers that were more than what we saw. And studies have also looked at pulling a lipid molecule through. So we might have like a lipid flip-flop across the membrane. Those also have considerable free energy barriers, more than what we're seeing. In some cases, and I've also seen cases where it's molecules that were different in chemical composition, but with lower free energy barriers. So I think the expected value of this is reasonable.
Does the sign of the charge make a difference?
Oh, the sign of the charge. I haven't seen much work on negatively charged compounds, and I imagine that might be more difficult in terms of membrane permeation, because these POPC molecules have the positively charged choline, which are sort of hydrated by the waters, but the negatively charged phosphates groups are more intermediate between the water and the membrane, so it actually might have a harder time permeating than the positively charged ligands.
I think it's the other way around, but I liked your reasoning. For negatively charged compounds, actually, the barrier is a bit smaller. May I ask about the last part of your talk?
Yeah, of course.
So you're using, to evaluate these force fields performance, you're using quantum mechanical calculations. So can you first tell me what level of quantum mechanical theory you're using?
Yeah, it's B3-lip with the D3-BJ correction, and then the DZVP basis set.
Okay. And as you know, all quantum mechanical methods are approximate, and there are some that are nearly exact for small molecules. Have you ever benchmarked, or are you aware of benchmarks of the quantum mechanical method that you used against super high quality, for example, coupled clusters up to triples, and in the complete basis set limit?
Yeah, so there's been a number of papers that have looked at this over time. So looking at various observables. And this actually led to a very robust discussion between the open force field researchers as they're trying to decide what level of theory we want to use, because this is, like I said, over 26,000 compounds, so we want to make sure we have a pretty reasonable reference. And so B3-lip with this basis set was chosen because it did a pretty good job of reproducing the conformer geometries and energies in the various literatures, as well as in some internal studies that they looked at. And with the DZVP basis set, it was a balance between the cost and the accuracy. So this was a good way for us to get reasonably good energies and geometries without running for days and days like we would have to do for a complete basis set calculation.
Great. And I have another question on that part of the talk as well. So typically when force fields for biomolecules are produced, there are multiple stages and different approaches, but oftentimes the starting point, of course, is to get the internal degrees of freedom all worked out by a comparison with quantum mechanical data. And then when you want to actually go to the next phase, you would typically use some condensed phase calculations to do refinements. Mm-hmm. And so what that means is that the quantum mechanical part is extremely important, but it's not the end of the game. So are any of these force fields that you're benchmarking, do they go through these additional stages of refinement? And if so, might that make it difficult to really use just purely quantum mechanical calculations to evaluate their fidelity?
Yeah, and that's a good question. Some of the force fields that I looked at in terms of their origin papers do report some condensed phase explorations of the different properties. And so we're using these in gas phase optimizations, and the idea behind that is that we don't expect that the valence properties will change very much between gas phase and condensed phase, so that we think we should be able still to reproduce those geometries correctly. And it's possible that the charges will play a role, because in gas phase, the effect of the charges might be a little bit stronger, which can lead to differences in conformations. But we don't think that should really affect the valence parameters that much.
Well, in condensed phase polarization, if it's a polar solvent like water, would definitely could significantly alter the charges. So, for example, if you were to take a quantum mechanical calculation for water and use the charges that you got out of that in the gas phase, it would never condense into a liquid. So polarization effects are important, and presumably would influence, to some extent at least, the torsional barriers, I guess. But so I guess the point I'm trying to make is that I think that exercise is really great, but it is complicated if the force fields have undergone a second round of optimization to take into consideration condensed phase properties.
Mm-hmm, certainly, yeah, and this is certainly not. These metrics are not saying this force field is certainly better. This is one aspect of the force field that we're looking at.
So I haven't gotten any questions coming into the chat, so I think we're all set.
Yeah, I have a couple of additional questions, but they can wait for the private session.
Okay, so this is when Vicky and the committee will jump over to that, and you should have a link to that a couple of times in your email, and then other people can stay here and eventually will reappear. Thanks.
Okay.
Thank you.
Sorry, is the email to that